14.5.2 Mechanical control. Mechanical control methods for keeping the housefly under control are 1. Screening or covering of windows, doors and trash containers proves useful in preventing breeding. 2. Fly traps, such as sticky traps, ultraviolet light traps and pheromone traps placed at different places in homes or in business places can reduce housefly population. Sticky traps can be prepared by coating a suitable device, flypaper with honey, sugar, fruit, molasses or any substance that attracts housefly. These flypaper devices are hanged in infested areas where there is little or no air movement and are dark. It is advisable to place one 10-inch strip per 1,000 cubic feet of space within 6 feet of the floor because most fly activity is near the ground and should be placed in dark areas away from bright light. Care should also be taken to place these traps away from the food and kitchen area to avoid the risk of food contamination. The strips are generally effective for up to about 3 months. B. Ultraviolet traps have a grid of wires mounted in front of the UV fluorescent tubes. The UV light attracts the files, trap and kill by electrocuting them. C. The sex pheromone Z-9-trichosan, called as muscalure, functions as an aggregation pheromone. Muscalure is added with sugar as a commercially available fly bait for local population suppression, as 14.5.3 Biological control. Parasitic wasps and fire ants suppress housefly populations naturally. The parasitic wasps do not harm people or animals. They kill immature houseflies. However, parasitic wasps alone cannot eliminate a housefly population. This method can be used in combination with other methods. 14.5.4 Chemical control. Insecticides can help suppress housefly populations. Fly baits, such as quickbait and golden marlin, are usually sugar-based and contain a compound that attracts the adult flies. Flies that feed on these baits are killed by the insecticide. Neem leaves and carbolic acid are good repellents for controlling flies. Fumigation can be done with dry neem leaves or carbolic acid. Repellents like creosote oil and borax solution sprinkled in animal sheds and other breeding places of the housefly are effective in controlling the fly population. Spraying of 2% malathion or diazinon, 1% chlordane or 0.5% dieldrin are effective insecticides for killing the flies. Anti-fly candles can be prepared by mixing pyrethrum, sawdust, wheat flour and potassium nitrate in the ratio of 5:1:2 2 and rolled into a candle. Burning of anti-fly candles gives a quick knockdown effect to the flies. Manure can also be treated with an insecticide, though this method is highly discouraged as it can be lethal to the biological control agents of flies. The housefly possesses sponging type of mouth parts to suck the liquid food. Adult body is divisible into 3 distinct regions viz. Head, thorax and abdomen. Hindwings are greatly reduced to form halteres which act as balancing organs. Glandular pads, pulvillus, are present on the legs in between the claws which enable the fly to walk on the walls or ceilings. Males have edagus, the copulatory organ, on the 9th abdominal segment. Ovipositor of female receives the sperms during copulation and help in egg laying. Development is indirect with complete metamorphosis, holometabolous. Eggs are white in color, elongated with a curved dorsal thickening. Larval form, called maggot, is legless, headless and passes through 3 instars. The first and second instar larvae feed on organic matter and debris voraciously. Pupa is enclosed in a pupal case called puparium and emerges into adult within 3 days. Adult flies generally do not live longer than 15 days in summers and 25 days in winters. The fly overwinters in the larval stage or pupal stage. Nutrient-rich substrates provide an excellent developmental substrate for the flies where they get warmth, sufficient moisture, nutrients and protection. Houseflies rely on reflected sunlight to detect objects in their environment while flying, finding food, searching for harborage and resting areas. The housefly is a mechanical vector of many pathogens: virus, bacteria, protists, fungi and nematodes. It is a potential mechanical vector for diseases like typhoid, paratyphoid fever, cholera, gastroenteritis, amoebiasis, salmonellosis, diarrhea, dysentery, anthrax, tuberculosis, trachoma, conjunctivitis and yaws. Eggs and larvae of tapeworm Taenia and roundworm Ascaris may be transmitted by housefly. Contact transmission, vomit drop and defecation are 3 different ways in which housefly can transmit the diseases. Laying of eggs on or in human tissues can cause myiasis, in which larvae. Eggs and larvae ingested with contaminated food and water can cause intestinal disorders. Mechanical, cultural, biological and chemical control methods can be used to keep a check on the housefly population. The integrated approach provides best result to control their population. 14.7 Fly. Flies are insects of the order Diptera, the name being derived from the Greek dd, 2, and pteron, wing. Insects of this order use only a single pair of wings to fly, the hindwings having evolved into advanced mechanosensory organs known as halteres, which act as high-speed sensors of rotational movement and allow dipterans to perform advanced aerobatics. Diptera is a large order containing an estimated 1 million species including horse flies, crane flies, hoverflies, mosquitoes and others, although only about 125,000 species have been described. An Anthemiidae species showing characteristic dipteran features: large eyes, small antennae, sucking mouthparts, single pair of flying wings, hindwings reduced to club-like halteres. Flies have a mobile head, with a pair of large compound eyes, and mouthparts designed for piercing and sucking. Mosquitoes, black flies and robber flies, or for lapping and sucking in the other groups. Their wing arrangement gives them great maneuverability in flight, and claws and pads on their feet enable them to cling to smooth surfaces. Flies undergo complete metamorphosis. The eggs are often laid on the larval food source, and the larvae, which lack true limbs, develop in a protected environment, often inside their food source. Other species are ovoviviparous, opportunistically depositing hatched or hatching larvae instead of eggs on carrion, dung, decaying material, or open wounds of mammals. The pupa is a tough capsule from which the adult emerges when ready to do so. Flies mostly have short lives as adults. Diptera is one of the major insect orders and of considerable ecological and human importance. Flies are important pollinators, second only to the bees and their hymenopteran relatives. Flies may have been among the evolutionarily earliest pollinators responsible for early plant pollination. Fruit flies are used as model organisms in research, but less benignly. Mosquitoes are vectors for malaria, dengue, West Nile fever, yellow fever, encephalitis, and other infectious diseases, and houseflies, commensal with humans all over the world, spread foodborne illnesses. Flies can be annoyances, especially in some parts of the world where they can occur in large numbers, buzzing and settling on the skin or eyes to bite or seek fluids. Larger flies such as tsetse flies and screwworms cause significant economic harm to cattle. Blowfly larvae, known as gentles, and other dipteran larvae, known more generally as maggots, are used as fishing bait, as food for carnivorous animals, and in medicine in debridement, to clean wounds. Relationships to other insects. Dipterans are holometabolans, insects that undergo radical metamorphosis. They belong to the Mecopteridae, alongside the Mecoptera, Siphonoptera, Lepidoptera and Trichoptera. 3. The possession of a single pair of wings distinguishes most true flies from other insects with fly in their names. However, some true flies such as Hippobosidae, louse flies, have become secondarily wingless. 5, 6. The cladogram represents the current consensus view. 7. Fly. Temporal range, Middle Triassic, recent. Diptera from different families. Housefly, Mussidae, top left, Hematopoda pluvialis, Tabanidae, top. Right, Ctenophora pectinicornis, Tipulidae, mid. Left, Oclerotatus notoscriptus, Culicidae. Mid-right, Milesia crabroniformis, Syrphidae. Bottom left, Holcocephala fusca, Acylidae, bottom right. Scientific classification domain, Eukaryota kingdom, Animalia phylum, Arthropoda class, Insecta superorder, Panorpoda. Taxonomy and phylogeny. Relationships between subgroups and families. Fossil brachyceran in Baltic amber. Lower Eocene, circa 50 million years ago. The first true dipterans known are from the Middle Triassic, around 240 million years ago, and they became widespread during the Middle and Late Triassic. 8 modern flowering plants did not appear until the Cretaceous, around 140 million years ago, so the original dipterans must have had a different source of nutrition other than nectar. Based on the attraction of many modern fly groups to shiny droplets, it has been suggested that they may have fed on honeydew produced by sap-sucking bugs which were abundant at the time, and dipteran mouthparts are well adapted to softening and lapping up the crusted residues. 9. The basal clades in the Diptera include the Deuterophlebiidae and the enigmatic Nymphomyidae. 10. 3 episodes of evolutionary radiation are thought to have occurred based on the fossil record. Many new species of lower Diptera developed in the Triassic, about 220 million years ago. Many lower Brachycera appeared in the Jurassic, some 180 million years ago. A third radiation took place among the Schiztophora at the start of the Paleogene, 66 million years ago. 10. The phylogenetic position of Diptera has been controversial. The monophily of holometabolous insects has long been accepted, with the main orders being established as Lepidoptera, Coleoptera, Hymenoptera and Diptera, and it is the relationships between these groups which has caused difficulties. Diptera is widely thought to be a member of Mecopteridae, along with Lepidoptera, butterflies and moths, Trichoptera, caddisflies, Siphonoptera, fleas, Mecoptera, scorpionflies, and possibly Strepsiptera, twisted wing flies. Diptera has been grouped with Siphonoptera and Mecoptera in the Antliaphora, but this has not been confirmed by molecular studies. 11. Fossil nematoceran in Dominican amber. Sandfly, Lutsomia adicetus, Psychotidae, early Miocene, circa 20 million years ago. Diptera were traditionally broken down into two suborders, Nematocera and Brachycera, distinguished by the differences in antennae. The Nematocera are identified by their elongated bodies and many segmented, often feathery antennae as represented by mosquitoes and crane flies. The Brachycera have rounder bodies and much shorter antennae. 12-13 Subsequent studies have identified the Nematocera as being non-monophyletic, with modern phylogenies placing the Brachycera within grades of groups formerly placed in the Nematocera. The construction of a phylogenetic tree has been the subject of ongoing research. The following cladogram is based on the Fly Tree project. 14-15. Blepharoceromorpha, net-winged midges, etc. Bibionomorpha, gnats. Psychotomorpha, drain flies, sand flies, etc. Tipulomorpha, crane flies. Abbreviations used in the cladogram: Cal equals Calyptratae. CYC equals Cyclorpha. Air equals Aeromonera. Muse equals Muscomorpha. SCH equals Schiztophora. Diversity. Goromidas heros is the largest fly in the world. Flies are often abundant and are found in almost all terrestrial habitats in the world apart from Antarctica. They include many familiar insects such as houseflies, blowflies, mosquitoes, gnats, black flies, midges and fruit flies. More than 150,000 have been formally described, and the actual species diversity is much greater, with the flies from many parts of the world yet to be studied intensively. 17. The suborder Nematocera include generally small, slender insects with long antennae such as mosquitoes, gnats, midges and crane flies, while the Brachycera includes broader, more robust flies with short antennae. Many Nematoceran larvae are aquatic. 18. There are estimated to be a total of about 19,000 species of Diptera in Europe, 22,000 in the Nearctic region, 20,000 in the Afrotropical region, 23,000 in the Oriental region and 19,000 in the Australasian region. 19. While most species have restricted distributions, a few like the housefly Musca domestica are cosmopolitan. 20. Goromydus heros Acyloidea with a length of up to 7 cm 2.8 in is generally considered to be the largest fly in the world. 21. While the smallest is Euryplata nonanile, which at 0.4 mm 0.016 in is smaller than a grain of salt. 22. Brachycera are ecologically very diverse, with many being predatory at the larval stage and some being parasitic. Animals parasitized include mollusks, woodlice, millipedes, insects, mammals, 19, and amphibians. 23. Flies are the second largest group of pollinators after the Hymenoptera, bees, wasps and relatives. In wet and colder environments, flies are significantly more important as pollinators. Compared to bees, they need less food as they do not need to provision their young. Many flowers that bear low nectar and those that have evolved trap pollination depend on flies. 24. It is thought that some of the earliest pollinators of plants may have been flies. 25. The greatest diversity of gall-forming insects are found among the flies, principally in the family Cystidomiidae, gall midges. 26. Many flies, most importantly in the family Agromyzidae, lay their eggs in the mesophyll tissue of leaves with larvae feeding between the surfaces forming blisters and mines. 27. Some families are mycophagous or fungus feeding. These include the cave-dwelling Mystophilidae, fungus gnats, whose larvae are the only Diptera with bioluminescence. The Charidae are also fungus feeders. Some plants are pollinated by fungus-feeding flies that visit fungus-infected male flowers. 28. The larvae of Megacelia scalaris, Phoridae, are almost omnivorous and consume such substances as paint and shoe polish. 29. The Ixoristomela, walker, fly are considered generalists and parasitoids of a variety of hosts. 30. The larvae of the shore flies, Ephydridae, and some Chironomidae survive in extreme environments including glaciers, Diamesa sp. Chironomidae 31, hot springs, geysers, saline pools, sulfur pools, septic tanks and even crude oil, Heliumia petrolae 31. 19. Adult hoverflies, Syrphidae, are well known for their mimicry and the larvae adopt diverse lifestyles including being inquiline scavengers inside the nests of social insects. 32. Some brachycerans are agricultural pests, some bite animals and humans and suck their blood, and some transmit diseases. 19. Anatomy and morphology. Flies are adapted for aerial movement and typically have short and streamlined bodies. The first tagma of the fly, the head, bears the eyes, the antennae, and the mouthparts, the labrum, labium, mandible, and maxilla make up the mouthparts. The second tagma, the thorax, bears the wings and contains the flight muscles on the second segment, which is greatly enlarged. The first and third segments have been reduced to collar-like structures, and the third segment bears the halteres, which help to balance the insect during flight. The third tagma is the abdomen consisting of 11 segments, some of which may be fused, and with the 3 hindmost segments modified for reproduction. 33-34. Some dipterans are mimics and can only be distinguished from their models by very careful inspection. An example of this is Spilomia longicornis, which is a fly but mimics a vespid wasp. Head of a horsefly showing large compound eyes and stout piercing mouthparts. A head of a fly, showing the 2 compound eyes and 3 simple eyes clearly. Flies have a mobile head with a pair of large compound eyes on the sides of the head, and in most species, 3 small ocelli on the top. The compound eyes may be close together or widely separated, and in some instances are divided into a dorsal region and a ventral region, perhaps to assist in swarming behavior. The antennae are well developed but variable, being thread-like, feathery or comb-like in the different families. The mouthparts are adapted for piercing and sucking, as in the black flies, mosquitoes and robber flies, and for lapping and sucking as in many other groups. 34. Female horse flies use knife-like mandibles and maxillae to make a cross-shaped incision in the host's skin and then lap up the blood that flows. The gut includes large diverticuli, allowing the insect to store small quantities of liquid after a meal. 35. For visual course control, flies' optic flow field is analyzed by a set of motion-sensitive neurons. 36. A subset of these neurons is thought to be involved in using the optic flow. 36. A subset of these neurons is thought to be involved in using the optic flow to estimate the parameters of self-motion, such as yaw, roll, and sideward translation. 37. Other neurons are thought to be involved in analyzing the content of the visual scene itself, such as separating figures from the ground using motion parallax. 38-39. The H1 neuron is responsible for detecting horizontal motion across the entire visual field of the fly, allowing the fly to generate and guide stabilizing motor corrections mid-flight with respect to yaw. 40. The ocelli are concerned in the detection of changes in light intensity, enabling the fly to react swiftly to the approach of an object. 41. Like other insects, flies have chemoreceptors that detect smell and taste, and mechanoreceptors that respond to touch. The third segments of the antennae and the maxillary palps bear the main olfactory receptors, while the gustatory receptors are in the labium, pharynx, feet, wing margins and female genitalia. 42. Enabling flies to taste their food by walking on it. The taste receptors in females at the tip of the abdomen receive information on the suitability of a site for ovipositing. 41. Flies that feed on blood have special sensory structures that can detect infrared emissions and use them to home in on their hosts. Many blood-sucking flies can detect the raised concentration of carbon dioxide that occurs near large animals. 43. Some tachnid flies, Ormianae, which are parasitoids of bush crickets, have sound receptors to help them locate their singing hosts. 44. A crane fly, showing the hind wings reduced to drumstick-shaped halteres. Diptera have one pair of forewings on the mesothorax and a pair of halteres, or reduced hind wings, on the metathorax. A further adaptation for flight is the reduction in number of the neural ganglia and concentration of nerve tissue in the thorax, a feature that is most extreme in the highly derived Muscomorpha infraorder. 35. Some flies such as the ectoparasitic Nycterebiidae and Striglidae are exceptional in having lost their wings and become flightless. The only other order of insects bearing a single pair of true, functional wings, in addition to any form of halteres, are the Strepsiptera. In contrast to the flies, the Strepsiptera bear their halteres on the mesothorax and their flight wings on the metathorax. 45. Each of the fly's 6 legs has a typical insect structure of coxa, trochanter, femur, tibia and tarsus, with the tarsus in most instances being subdivided into 5 tarsomers. 34. At the tip of the limb is a pair of claws, and between these are cushion-like structures known as pulvuli which provide adhesion. 46. The abdomen shows considerable variability among members of the order. It consists of 11 segments in primitive groups and 10 segments in more derived groups, the 10th and 11th segments having fused. 47. The last 2 or 3 segments are adapted for reproduction. Each segment is made up of a dorsal and a ventral sclerote, connected by an elastic membrane. In some females, the sclerotes are rolled into a flexible, telescopic ovipositor. 34. Flight. Tabanid fly in flight. Flies are capable of great maneuverability during flight due to the presence of the halteres. These act as gyroscopic organs and are rapidly oscillated in time with the wings. They act as a balance and guidance system by providing rapid feedback to the wing steering muscles, and flies deprived of their halteres are unable to fly. The wings and halteres move in synchrony, but the amplitude of each wing beat is independent, allowing the fly to turn sideways. 48. The wings of the fly are attached to 2 kinds of muscles, those used to power it and another set used for fine control. 49. Flies tend to fly in a straight line, then make a rapid change in direction before continuing on a different straight path. The directional changes are called saccades and typically involve an angle of 90 degrees, being achieved in 50 milliseconds. They are initiated by visual stimuli as the fly observes an object. Nerves then activate steering muscles in the thorax that cause a small change in wing stroke which generates sufficient torque to turn. Detecting this within 4 or 5 wing beats, the halteres trigger a counter turn and the fly heads off in a new direction. 50. Flies have rapid reflexes that aid their escape from predators, but their sustained flight speeds are low. Dolichopodid flies in the genus Condylostylus respond in less than 5 milliseconds to camera flashes by taking flight. 51. In the past, the deerbot fly, Cephemia, was claimed to be one of the fastest insects on the basis of an estimate made visually by Charles Townsend in 1927. 52. This claim, of speeds of 600 to 800 miles per hour, was regularly repeated until it was shown to be physically impossible as well as incorrect by Irving Langmuir. Langmuir suggested an estimated speed of 25 miles per hour. 53, 54, 55. Although most flies live and fly close to the ground, a few are known to fly at heights, and a few like Ocinella chloropody are known to be dispersed by winds at altitudes of up to 2,000 feet and over long distances. 56. Some hoverflies like Metacerphus corolli have been known to undertake long flights in response to aphid population spurts. 57. Males of fly species such as Cuterbra, many hoverflies, 58 bee flies Bombylidae, 59, and fruit flies Tephrididae, 60, maintain territories within which they engage in aerial pursuit to drive away intruding males and other species. 61. While these territories may be held by individual males, some species, such as a freeborni, 62, form leks with many males aggregating in displays. 60. Some flies maintain an airspace and still others form dense swarms that maintain a stationary location with respect to landmarks. Many flies mate in flight while swarming. 63. Life cycle and development. Mating anthemiid flies. Diptera go through a complete metamorphosis with 4 distinct life stages: egg, larva, pupa and adult. Larva. In many flies, the larval stage is long and adults may have a short life. Most dipteran larvae develop in protected environments. Many are aquatic and others are found in moist places such as carrion, fruit, vegetable matter, fungi and, in the case of parasitic species, inside their hosts. They tend to have thin cuticles and become desiccated if exposed to the air. Apart from the Brachycera, most dipteran larvae have sclerotized head capsules, which may be reduced to remnant mouth hooks. The Brachycera, however, have soft, gelatinized head capsules from which the sclerites are reduced or missing. Many of these larvae retract their heads into their thorax. 34, 64. The spiracles in the larva and pupa do not have any internal mechanical closing device. 65. Life cycle of stable fly Stomoxys calcitrans, showing eggs, 3 larval instars, pupa, and adult. Some other anatomical distinction exists between the larvae of the Nematocera and the Brachycera. Especially in the Brachycera, little demarcation is seen between the thorax and abdomen, though the demarcation may be visible in many Nematocera, such as mosquitoes. In the Brachycera, the head of the larva is not clearly distinguishable from the rest of the body, and few, if any, sclerites are present. Informally, such brachyceran larvae are called maggots. 66. But the term is not technical and often applied indifferently to fly larvae or insect larvae in general. The eyes and antennae of brachyceran larvae are reduced or absent, and the abdomen also lacks appendages such as cerci. This lack of features is an adaptation to food such as carrion, decaying detritus, or host tissues surrounding endoparasites. 35. Nematoceran larvae generally have well-developed eyes and antennae, while those of brachyceran larvae are reduced or modified. 67. Dipteran larvae have no jointed, true legs. 64. But some dipteran larvae, such as species of Simuliidae, Tabanidae and Vermilionidae, have prolegs adapted to hold onto a substrate in flowing water, host tissues or prey. 68. The majority of dipterans are oviparous and lay batches of eggs, but some species are ovoviviparous, where the larva starting development inside the eggs before they hatch are viviparous, the larva hatching and maturing in the body of the mother before being externally deposited. These are found especially in groups that have larvae dependent on food sources that are short-lived or are accessible for brief periods. 69. This is widespread in some families such as the Sarcophagidae. In Hyalmia strigosa, Entomology, the larva molts to the second instar before hatching, and in Termitoxenia phoridae, females have incubation pouches, and a full-developed third instar larva is deposited by the adult and it almost immediately pupates with no freely feeding larval stage. The tsetse fly, as well as other Glossinidae, Hippobosidae, Nycteribidae and Striglidae, exhibits a denatrophic viviparity. A single fertilized egg is retained in the oviduct and the developing larva feeds on glandular secretions. When fully grown, the female finds a spot with soft soil and the larva works its way out of the oviduct, buries itself and pupates. Some flies like Lunstroemia parthenogenetica, Chironomidae, reproduce by Thelytocus parthenogenesis, and some gall midges have larvae that can produce eggs, paedogenesis. 71. Pupa. The pupae take various forms. In some groups, particularly the Nematocera, the pupa is intermediate between the larval and adult form. These pupae are described as obtect, having the future appendages visible as structures that adhere to the pupal body. The outer surface of the pupa may be leathery and bear spines. Respiratory features are locomotory paddles. In other groups, described as coarctate, the appendages are not visible. In these, the outer surface is a puparium, formed from the last larval skin, and the actual pupa is concealed within. When the adult insect is ready to emerge from this tough, desiccation-resistant capsule, it inflates a balloon-like structure on its head, and forces its way out. 34. Adult. The adult stage is usually short. Its function is only to mate and lay eggs. The genitalia of male flies are rotated to a varying degree from the position found in other insects. 72. In some flies, this is a temporary rotation during mating, but in others, it is a permanent torsion of the organs that occurs during the pupal stage. This torsion may lead to the anus being below the genitals, or, in the case of 360 degrees torsion, to the sperm duct being wrapped around the gut and the external organs being in their usual position. When flies mate, the male initially flies on top of the female, facing in the same direction, but then turns around to face in the opposite direction. This forces the male to lie on his back for his genitalia to remain engaged with those of the female, or the torsion of the male genitals allows the male to mate while remaining upright. This leads to flies having more reproduction abilities than most insects, and much quicker. Flies occur in large populations due to their ability to mate effectively and quickly during the mating season. 35. More primitive groups mates in the air during swarming, but most of the more advanced species with a 360 degrees torsion mate on a substrate. 73. Ecology. A caliphorid, bubbling. As ubiquitous insects, dipterans play an important role at various trophic levels both as consumers and as prey. In some groups the larvae complete their development without feeding, and in others the adults do not feed. The larvae can be herbivores, scavengers, decomposers, predators or parasites, with the consumption of decaying organic matter being one of the most prevalent feeding behaviors. The fruit or detritus is consumed along with the associated microorganisms, a sieve-like filter in the pharynx being used to concentrate the particles, while flesh-eating larvae have mouth hooks to help shred their food. The larvae of some groups feed on or in the living tissues of plants and fungi, and some of these are serious pests of agricultural crops. Some aquatic larvae consume the films of algae that form underwater on rocks and plants. Many of the parasitoid larvae grow inside and eventually kill other arthropods, while parasitic larvae may attack vertebrate hosts. 34. Whereas many dipteran larvae are aquatic or live in enclosed terrestrial locations, the majority of adults live above ground and are capable of flight. Predominantly they feed on nectar or plant or animal exudates, such as honeydew, for which their lapping mouthparts are adapted. Some flies have functional mandibles that may be used for biting. The flies that feed on vertebrate blood have sharp stylets that pierce the skin, with some species having anticoagulant saliva that is regurgitated before absorbing the blood that flows. In this process, certain diseases can be transmitted. The bot flies, Estridae, have evolved to parasitize mammals. Many species complete their life cycle inside the bodies of their hosts. 74. The larvae of a few fly groups, Agromizidae, Anthemiidae, Cycidomiidae, are capable of inducing plant galls. Some dipteran larvae are leaf miners. The larvae of many brachyceran families are predaceous. In many dipteran groups, swarming is a feature of adult life, with clouds of insects gathering in certain locations. These insects are mostly males, and the swarm may serve the purpose of making their location more visible to females. 34. Most adult diptera have their mouthparts modified to sponge up fluid. The adults of many species of flies, e.g. Anthemia sp. Steganopsis melanogaster, that feed on liquid food will regurgitate fluid in a behavior termed as bubbling, which has been thought to help the insects evaporate water and concentrate food. 75. Or possibly to cool by evaporation. 76. Some adult diptera are known for kleptoparasitism such as members of the Sarcophagidae. The Miltigraminae are known as satellite flies for their habit of following wasps and stealing their stung prey or laying their eggs into them. Phorids, milichids in the genus Bengalia are known to steal food carried by ants. 77. Adults of Ephydraheans forage underwater and have special hydrophobic hairs that trap a bubble of air that lets them breathe underwater. 78. Anti-predator adaptations. The large bee fly, Bombylius major, is a Batesian mimic of bees. Flies are eaten by other animals at all stages of their development. The eggs and larvae are parasitized by other insects and are eaten by many creatures, some of which specialize in feeding on flies but most of which consume them as part of a mixed diet. Birds, bats, frogs, lizards, dragonflies and spiders are among the predators of flies. 79. Many flies have evolved mimetic resemblances that aid their protection. Batesian mimicry is widespread with many hoverflies resembling bees and wasps, 80, 81, ants, 82, and some species of tephritid fruit fly resembling spiders. 83. Some species of hoverfly are myrmecophilous, their young live and grow within the nests of ants. They are protected from the ants by imitating chemical odors given by ant colony members. 84. Bombolid bee flies such as Bombylius major are short-bodied, round, furry, and distinctly bee-like as they visit flowers for nectar, and are likely also Batesian mimics of bees. 85. In contrast, Drosophila subobscura, a species of fly in the genus Drosophila, lacks a category of hemocytes that are present in other studied species of Drosophila, leading to an inability to defend against parasitic attacks, a form of innate immunodeficiency. 86. Human interaction and cultural depictions. Symbolism. Petrus Christus's 1446 painting portrait of a Carthusian has a musca depicta, painted fly, on a trompe-l'oeil frame. Flies play a variety of symbolic roles in different cultures. These include both positive and negative roles in religion. In the traditional Navajo religion, Big Fly is an important spirit being. 87, 88, 89. In Christian demonology, Beelzebub is a demonic fly, the Lord of the Flies, and a god of the Philistines. 90, 91, 92. Flies have appeared in literature since ancient Sumer. 93. In a Sumerian poem, a fly helps the goddess Inanna when her husband Dumuzid is being chased by gala demons. 93. In the Mesopotamian versions of the flood myth, the dead corpses floating on the waters are compared to flies. 93. Later, the gods are said to swarm, like flies, around the hero Unapishtim's offering. 93. Flies appear on Old Babylonian seals as symbols of Nergal, the god of death. 93. Fly-shaped lapis lazuli beads were often worn in ancient Mesopotamia, along with other kinds of fly jewelry. 93. In ancient Egypt, flies appear in amulets and as a military award for bravery and tenacity, due to the fact that they always come back when swatted at. It is thought that flies may have also been associated with the departing spirit of the dead, as they are often found near dead bodies. In modern Egypt, a similar belief persists in some areas to not swat at shiny green flies, as they may be carrying the soul of a recently deceased person. 94. In a little-known Greek myth, a very chatty and talkative maiden named Maia, meaning fly, enraged the moon goddess Selene by attempting to seduce her lover, the sleeping Endymion, and was thus turned by the angry goddess into a fly, who now always deprives people of their sleep in memory of her past life. 95, 96. In Prometheus Bound, which is attributed to the Athenian tragic playwright Aeschylus, a gadfly sent by Zeus's wife Hera pursues and torments his mistress Io, who has been transformed into a cow and is watched constantly by the 100 eyes of the herdsman Argus. 97, 98. Io. Ah. Ha. Again the prick, the stab of gadfly sting. O earth, earth, hide, the hollow shape. Argus, that evil thing, the 100-eyed. 98. William Shakespeare, inspired by Aeschylus, has Tom o' Bedlam in King Lear, whom the foul fiend hath led through fire and through flame, through ford and whirlpool, o'er bog and quagmire, driven mad by the constant pursuit. 98. In Antony and Cleopatra, Shakespeare similarly likens Cleopatra's hasty departure from the Actium battlefield to that of a cow chased by a gadfly. 99. More recently, in 1962 the biologist Vincent de Thier wrote To Know a Fly, introducing the general reader to the behavior and physiology of the fly. 100. Musca depicta, painted fly, in Latin, is a depiction of a fly as an inconspicuous element of various paintings. This feature was widespread in 15th and 16th centuries paintings and its presence may be explained by various reasons. 101. Flies appear in popular culture in concepts such as fly-on-the-wall documentary making in film and television production. The metaphoric name suggests that events are seen candidly, as a fly might see them. 102. Flies have inspired the design of miniature flying robots. 103. Steven Spielberg's 1993 film Jurassic Park relied on the idea that DNA could be preserved in the stomach contents of a blood-sucking fly fossilized in amber, though the mechanism has been discounted by scientists. 104. Economic importance. An Anopheles stevensii mosquito drinking human blood. The species carries malaria. Dipterans are an important group of insects and have a considerable impact on the environment. Some leaf miner flies, Agromyzidae, fruit flies, Tephrididae and Drosophilidae, and gall midges, Cecidomyidae, are pests of agricultural crops. Others such as tsetse flies, screwworm and botflies, Estridae, attack livestock, causing wounds, spreading disease, and creating significant economic harm. See article, Parasitic flies of domestic animals. A few can even cause myiasis in humans. Still others such as mosquitoes, Culicidae, blackflies, Simuliidae, and drain flies, Psychotidae, impact human health, acting as vectors of major tropical diseases. Among these, Anopheles mosquitoes transmit malaria, filariasis, and arboviruses. Aedes aegypti mosquitoes carry dengue fever and the Zika virus. Black flies carry river blindness. Sand flies carry leishmaniasis. Other dipterans are a nuisance to humans, especially when present in large numbers. These include houseflies, which contaminate food and spread foodborne illnesses, the biting midges and sandflies, Ceratopogonidae, and the houseflies and stable flies, Mucidae. 34. In tropical regions, eye flies, Chloropodidae, which visit the eye in search of fluids, can be a nuisance in some seasons. 105. Many dipterans serve roles that are useful to humans. Houseflies, blowflies and fungus gnats, Mystophilidae, are scavengers and aid in decomposition. Robber flies, Acylidae, Tachinids, Tachinidae, and dagger flies and balloon flies, Empedidae, are predators and parasitoids of other insects, helping to control a variety of pests. Many dipterans such as bee flies, Bombylidae, and hoverflies, Syrphidae, are pollinators of crop plants. 34. Uses. Diptera in research. Drosophila melanogaster fruit fly larva being bred in tubes in a genetics laboratory. Drosophila melanogaster, a fruit fly, has long been used as a model organism in research because of the ease with which it can be bred and reared in the laboratory, its small genome, and the fact that many of its genes have counterparts in higher eukaryotes. A large number of genetic studies have been undertaken based on this species. These have had a profound impact on the study of gene expression, gene regulatory mechanisms, and mutation. Other studies have investigated physiology, microbial pathogenesis, and development among other research topics. 106. The studies on dipteran relationships by Willy Hennig helped in the development of cladistics, techniques that he applied to morphological characters but now adapted for use with molecular sequences in phylogenetics. 107. Maggots found on corpses are useful to forensic entomologists. Maggot species can be identified by their anatomical features and by matching their DNA. Maggots of different species of flies visit corpses and carcasses at fairly well-defined times after the death of the victim, and so do their predators, such as beetles in the family Hysteridae. Thus, the presence or absence of particular species provides evidence for the time since death, and sometimes other details such as the place of death, when species are confined to particular habitats such as woodland. 108. Casu marzu is a traditional Sardinian sheep milk cheese that contains larvae of the cheese fly, Piophilicaceae. Some species of maggots such as blowfly larvae, gentles, and bluebottle larvae, castors, are bred commercially. They are sold as bait in angling, and as food for carnivorous animals kept as pets, in zoos, or for research such as some mammals, 109 fishes, reptiles, and birds. It has been suggested that fly larvae could be used at a large scale as food for farmed chickens, pigs, and fish. However, consumers are opposed to the inclusion of insects in their food, and the use of insects in animal feed remains illegal in areas such as the European Union. 110. 111. Fly larvae can be used as a biomedical tool for wound care and treatment. Maggot debridement therapy, MDT, is the use of blowfly larvae to remove the dead tissue from wounds, most commonly being amputations. Historically, this has been used for centuries, both intentional and unintentional, on battlefields and in early hospital settings. 112. Removing the dead tissue promotes cell growth and healthy wound healing. The larvae also have biochemical properties such as antibacterial activity found in their secretions as they feed. 113. These medicinal maggots are a safe and effective treatment for chronic wounds. 114. The Sardinian cheese Casu marzu is exposed to flies known as cheese skippers such as Piophilicaceae, members of the family Piophilidae. 115. The digestive activities of the fly larvae soften the cheese and modify the aroma as part of the process of maturation. At one time European Union authorities banned sale of the cheese and it was becoming hard to find. 116. But the ban has been lifted on the grounds that the cheese is a traditional local product made by traditional methods. 117. Hazards. Flies are a health hazard and are attracted to toilets because of their smell. The New Scientist magazine suggested a trap for these flies. A pipe acting as a chimney was fitted to the toilet which let in some light to attract these flies up to the end of this pipe where a gauze covering prevented escape to the air outside so that they were trapped and died. Toilets are generally dark inside, particularly if the door is closed. Notes. A. Some authors draw a distinction in writing the common names of insects. True flies are in their view best written as two words, such as crane fly, robber fly, bee fly, moth fly, and fruit fly. In contrast, common names of non-dipteran insects that have fly in their names are written as one word, e.g. butterfly, stonefly, dragonfly, scorpionfly, sawfly, caddisfly, whitefly. 1. In practice, however, this is a comparatively new convention, especially in older books. Names like sawfly and caddisfly, or hyphenated forms such as housefly and dragonfly are widely used. 2. Exceptions to this rule occur, such as the hoverfly, which is a true fly, and the Spanish fly, a type of blister beetle. Forensic entomology. Forensic entomology is a branch of forensic science that uses insects found on corpses to help solve criminal cases. This includes studying the types of insects commonly found on cadavers, their life cycles, their presence in different environments, and how insect assemblages change with decomposition. 1. Insect succession patterns are identified based on the time a species spends in each developmental stage and the number of generations produced since the insect's introduction to a food source. 2. By analyzing insect development alongside environmental data such as temperature, humidity, and vapor density, forensic entomologists can estimate the time since death, as flying insects are attracted to a body immediately after death. This field also provides clues about body movement after death and antemortem trauma. 3. 4. The primary goal is to determine the postmortem interval, PMI, to aid in death investigations. However, forensic entomology is also used in cases of neglect and abuse as toxicology to detect drugs and food contamination incidents. Insect assemblages can help approximate a body's primary location, as some insects are unique to specific areas. 5. Therefore, forensic entomology is divided into 3 subfields: urban, stored product, and medico-legal, medico-criminal entomology. History. Historically, several accounts of applications have been for, and experimentation with, forensic entomology. Aside from an initial case report in China from the 13th century, the primitive observation and correlation between arthropods and forensic contexts has been documented in Germany and France. This observation was conducted during a mass exhumation in the late 1880s by Hoffman and Reinhard. 6. However, only in the last 30 years has forensic entomology been systematically explored as a feasible source for evidence in criminal investigations. Through documented experiments and focus on arthropods and death, the works of Sun Tzu, Francesco Redi, Bergeret d'Arbois, Jean-Pierre Megnin and the physiologist Hermann Reinhard form the foundations for today's modern forensic entomology. Song C. The Song Dynasty 960-1279 Forensic Science Book collected cases of injustice rectified published by a court judge, physician, medical scientist, and writer Song C in 1247 contains the oldest known case of forensic entomology. 7. In a murder case of 1235, a villager was stabbed to death and authorities determined that his wounds were inflicted by a sickle. This was a tool used for cutting rice at harvest time, a fact which led them to suspect a fellow peasant worker was involved. 7. The local magistrate had the villagers assemble in the town square, where they would temporarily relinquish their sickles. 7. Within minutes, a mass of blowflies gathered around one sickle and no others, attracted to the scent of traces of blood unseen by the naked eye. 7. It became apparent to all that the owner of that sickle was the culprit, the latter pleading for mercy as authorities detained him. 7. Song Si, sometimes called Sun Tzu, was a judicial intendant who lived in China from 1188 to 1251 AD. In 1247 AD, Song Si wrote a book entitled Washing Away of Wrongs as a coroner handbook. 8. In this book Song Si depicts several cases in which he took notes on how a person died and elaborates on probable cause. He explains in detail how to examine a corpse before and after burial. He also explains the process of how to determine a probable cause of death. The main purpose of this book was to be used as a guide for other investigators so they could assess the crime scene effectively. His level of detail in explaining what he observed in all his cases laid down the fundamentals for modern forensic entomologists. He is the first recorded account of someone using forensic entomology for judicial means. 9. Francesco Redi. In 1668, Italian physician Francesco Redi disproved the theory of spontaneous generation. This was the accepted theory of Redi's day, and it claimed that maggots developed spontaneously from rotting meat. In an experiment, Redi used samples of rotting meat that were either fully exposed to the air, partially exposed to the air, or not exposed to the air. Redi showed that both fully and partially exposed rotting meat developed fly maggots, whereas rotting meat not exposed to air did not develop maggots. This discovery completely changed how people viewed organisms' decomposition and prompted further investigations into insect life cycles and entomology in general. 10. Bergeret d'Arbois. Dr. Louis-François Étienne Bergeret, 1814-1893, was a French hospital physician and was the first to apply forensic entomology to a case. In a case report published in 1855, he stated a general life cycle for insects and made many assumptions about their mating habits. Nevertheless, these assumptions led him to the first application of forensic entomology in an estimation of postmortem interval (PMI). His report used forensic entomology as a tool to prove his hypothesis on how and when the person had died. 6. Hermann Reinhard. The first systematic study in forensic entomology was conducted in 1881 by Hermann Reinhard, a German medical doctor. He exhumed many bodies and demonstrated that the development of many different insect species could be tied to buried bodies. Reinhard conducted his first study in East Germany and collected many phorid flies there. He concluded that the development of only some of the insects living near corpses underground was associated directly with decaying flesh, since there were 15-year-old beetles who had little direct contact with the bodies. Reinhard's works and studies were used extensively in further forensic entomology studies. Jean-Pierre Megnin. French veterinarian and entomologist Jean-Pierre Megnin, 1828-1905, published many articles and books on various subjects, including the books Fond des Tombeaux and Le Fond des Cadavres, which are considered to be among the most important forensic entomology books in history. 11. In his second book he did revolutionary work on the theory of predictable waves, or successions of insects onto corpses. By counting numbers of live and dead mites every 15 days and comparing the data with his initial count on the infant, he was able to estimate how long the infant had been dead. 6. In this book, he asserted that exposed corpses were subject to 8 successional waves, whereas buried corpses were only subject to 2 waves. Megnin made many great discoveries that helped shed new light on many of the general characteristics of decaying flora and fauna. Megnin's work and study of the larval and adult forms of insect families found in cadavers sparked the interest of future entomologists and encouraged more research in the link between arthropods and the deceased, and thereby helped to establish the scientific discipline of forensic entomology. Forensic entomology subfields. Urban forensic entomology. Urban forensic entomology typically concerns pest infestations in buildings, gardens, or other urban environments, and may be the basis of litigation between private parties and service providers such as landlords or exterminators. 12. For instance, urban forensic entomology can be used to evaluate the efficiency of pest control techniques, ascertain the size of an infestation, and identify the responsible party in situations involving infestations in rental homes. Urban forensic entomology studies may also indicate the fitness of certain pesticide treatments. Urban forensic entomology can also assist in determining liability when stored goods, like grains or packaged foods, are contaminated with insects, helping to identify the infestation's origin. These techniques may be used in stored products cases where they can help to determine the chain of custody, when all points of possible infestation onset are examined to determine who is at fault. 13. Moreover, environmental management and public health depend heavily on urban forensic entomology. Researchers can track the transmission of disease carried by insects by examining insect populations in urban settings. Forensic techniques can also guide conservation efforts by evaluating the environmental effects of urbanization on insect populations. Stored product forensic entomology. Stored product forensic entomology is often used in litigation over insect infestation or contamination of commercially distributed foods, including grains, flour, and packaged meals. 12-14. Stored product forensic entomologists may be asked to identify the bug species involved, evaluate the extent of an infestation, and pinpoint the infestation's source in a legal proceeding. 15. They might offer expert testimony about the circumstances that gave rise to the infestation and suggest safeguards to prevent similar risks in the future. Forensic entomology on stored products both adds critical evidence to legal evaluation and contributes to overall food safety and quality assurance. Forensic entomologists work to guarantee that food products are safe for consumption by identifying insect species and tracking their presence in stored goods. Additionally, this sector contributes to the general enhancement of food business practices by researching and developing novel techniques for pest management and product preservation. 15. Medico-legal forensic entomology. Medico-legal forensic entomology involves the study of arthropods found at the scene of various incidents such as murder, suicide, rape, physical abuse and contraband trafficking. 12. Forensic investigators can learn important details from insect activities, including the length of time passed since death, the presence of medication or toxins in the body, and the movement or disturbance of the body following death. In murder investigations, forensic entomologists analyze which insects' eggs appear, their location on human remains, and their development stage to determine the PMI and the location of death. The presence of specific insect species, which may exhibit endemism, occurring only in certain places, or a well-defined phenology, active only at a certain season, or time of day, in association with other evidence can provide crucial links to times and locations where other criminal acts may have occurred. 16-17. This discipline provides techniques to associate a victim, suspect and scene together by identifying different insect species found in specific geographical locations. 18. Another area covered by medico-legal forensic entomology is the relatively new field of entomotoxicology. This particular branch involves testing entomological specimens found at a scene for different drugs that may have possibly played a role in the death of the victim. The analytical perspective behind these methods relies upon the fact that the presence of drugs within the carcass specifically affects the growth and morphology of the insects ingesting those toxins from the corpse. 19. Due to these alterations, the presence of drugs can potentially lead to an erroneous PMI when basing it on the abnormal physical development of insects who have fed on them. 20. Entomology can aid in medico-legal cases when determining the time of an injury. One determining factor might be the observed species preference of alimentation. When eggs laid on a corpse by blowflies subsequently hatch into first instar, first stage larvae, they require a liquid protein meal. 21. Due to their minute size and fragility, blowflies are unable to break through human skin themselves to attain this nutrition. Therefore, the female typically oviposits near a pre-existing wound or natural orifice to provide accessible blood, a mucosal layer and body fluids for her offspring to eat. Myiasis. Myiasis, the infestation of living vertebrate animals with dipteran larvae, blowfly maggots, is a phenomenon that may be noted in cases of abandonment or neglect. 21. This condition occurs when flies colonize a living human or animal, feeding on the organism's accessible living tissues, ingesting food or bodily fluids. 21. In a forensic context, myiasis can be confusing, as it may indicate the time of neglect or injury rather than PMI if the victim or remains were colonized when alive and prior to discovery. This highlights the importance of careful interpretation of all evidence in forensic investigation. 22-21. The role of insects in the decomposition processes. Insect activities are essential to the breakdown of organic materials, including human remains. Chemicals emitted during decomposition attract necrophilic insects, those that feed on dead creatures. These insects hasten the decomposition process by aiding in the breakdown of bodily tissues. 23. Among the first insects to reach a body, blowflies are classified as primary colonizers. Their oviposited eggs are laid in natural openings, wounds, or damp places, and decaying tissues are consumed by their maggot larvae. By feeding on the remnants, other arthropods like mites and beetles may also aid in the decomposition process. Invertebrate types. Scorpion flies. Scorpion flies, order Macoptera, were the first insects to arrive at a donated human cadaver observed by the entomologist Natalie Lindgren at the Southeast Texas Applied Forensic Science Facility near Huntsville, Texas, and remained on the corpse for 1.5 days, outnumbering flies during that period. A great presence of scorpion flies may indicate a shorter postmortem interval. 24-25. Flies. Flies from the order Diptera are often found at a crime scene, because they are attracted to the chemicals released by decomposing bodies, known as volatile organic compounds (VOCs). 26. A dead body is a perfect environment for oviposition, the laying of eggs. 26. A dead body is. Flies from the order Diptera are often found at a crime scene, because they are attracted to the chemicals released by decomposing bodies, known as volatile organic compounds (VOCs). 26. A dead body is a perfect environment for oviposition, the laying of eggs. The developing maggots will have a ready food source. Different types of flies can be found on dead bodies, the most significant of which are blowflies on a corpse. Blowflies, family Calliphoridae. In this family are often metallic in appearance and between 10 and 14 millimeters in length. 27. In addition to the name blowfly, some members of this family are known as blue bottle fly, cluster flies, green bottles, or black blowfly. A characteristic of the blowfly is its three-segmented antennae. Hatching from an egg to the first larval stage takes from 8 hours to 1 day. Larvae have 3 stages of development called instars. Each stage is separated by a molting event. Molting can be defined as the process of new cuticle production while subsequently shedding the old cuticle. 28. Larva's ideal habitat in regard to pupation are locations providing access to loose, damp soil and litter. The latter consists of temperate and rather tropical areas. 29. Worldwide, there are 1,100 known species of blowflies, with 228 species in the neotropics, and a large number of species in Africa and southern Europe. The most common area to find Calliphoridae species are in the countries of India, Japan, Central America, and in the southern United States. The forensic importance of this fly is that it is the first insect to come in contact with carrion because they have the ability to smell death from up to 10 miles, 16 kilometers away. 30. Some prominent species of Calliphoridae are Calliphora vomitoria and Calliphora vicina. Flesh fly on decomposing flesh. Flesh flies, family Sarcophagidae. Most flesh flies breed in carrion, dung, garbage, or decaying material, but a few species lay their eggs in the open wounds of mammals, hence their common name. Characteristics of the flesh fly is its 3-segmented antennae. Most Holarctic Sarcophagidae vary in size from 4 to 18 millimeters in length. Tropical species can be larger, with black and gray longitudinal stripes on the thorax and checkering on the abdomen. Flesh flies, being viviparous, frequently give birth to live young on corpses of human and other animals, at any stage of decomposition, from newly dead through to bloated or decaying, though the latter is more common. Sarcophaga barbata are specifically useful since they deposit maggots directly onto the decomposing body, their larger, visible size, and difference in activity during different stages. Their main limitation, however, is due to lack of information surrounding their geographic distribution and taxonomic features. Timeline of postmortem changes, stages of death, including housefly eggs, larvae and pupae. Housefly, family Muscidae, is the most common of all flies found in homes, and indeed one of the most widely distributed insects. It is often considered a pest that can carry serious diseases. The adults are 6 to 9 m long. Their thorax is gray, with 4 longitudinal dark lines on the back. The underside of their abdomen is yellow, and their whole body is covered with hair. Each female fly can lay up to 500 eggs in several batches of about 75 to 150 eggs. Genus Hydrotia are of particular forensic importance. Cheese flies, family Piophilidae. Most are scavengers in animal products and fungi. The best known member of the family is Piophilicaceae. It is a small fly, about 4 millimeters, 1/6 inch long, found worldwide. This fly's larva infests cured meats, smoked fish, cheeses, and decaying animals and is sometimes called the cheese skipper for its leaping ability. Forensic entomology uses the presence of Piophilicaceae larvae to help estimate the date of death for human remains. They do not take up residence in a corpse until 3 to 6 months after death. The adult fly's body is black, blue-black, or bronze, with some yellow on the head, antennae, and legs. The wings are faintly iridescent and lie flat upon the fly's abdomen when at rest. At 4 millimeters, 1/6 inch long, the fly is 1/3 to 1/2 as long as the common housefly. Coffin flies, Phoridae, aka humpbacked flies. Larvae feed on decaying bodies. Some species can burrow to a depth of 50 centimeters over 4 days. Important in buried bodies. Lesser corpse flies, Sphaeroceridae. Lesser house flies, Phaniidae. Black scavenger flies, Sepsidae. Sun flies, Heliomyzidae. Black soldier fly, Stratiomyidae, have potential for use in forensic entomology. The larvae are common scavengers in compost heaps, are found in association with carrion, can be destructive pests in honeybee hives, and are used in manure management for both housefly control and reduction in manure volume. The larvae range in size from 1/8 to 3/4 of an inch, 3 to 19 millimeters. The adult fly is a mimic, very close in size, color, and appearance to the organ pipe mud dauber wasp and its relatives. Non-biting midges Chironomidae. These flies have a complex life cycle. While adults are terrestrial and phytophagous, larvae are aquatic and detritivorous. Immature instars have been used as forensic markers in several cases where submerged corpses were found. 31. Beetles. Beetles, order Coleoptera, are generally found in later stages of decomposition. 32. They play a role in breaking down the remaining tissue and are important in the final stages of decomposition. In drier conditions, the beetles can be replaced by moth flies, Psychotidae. Their life cycle normally consists of 4 stages: eggs, larval, pupal, and adult. Each stage has unique eating behavior that changes as the organism breaks down. In forensic investigations, the existence and developmental phases of insects can yield important data for understanding environmental circumstances, body movement, and postmortem interval (PMI) estimation. In forensic entomology, insects play a crucial function as indicators, helping to identify vital components of a death investigation due to their distinct ecological responsibilities and varied geographic distribution. Different types of beetles can be found on dead bodies. The most significant include Rove beetle. Rove beetles, family Staphylinidae, are elongate beetles with small elytra, wing covers, and large jaws. Rove beetles have a 4-stage life cycle: egg, larva, pupa, and adult. 33. Criophilus species are common predators of carrion, and since they are large, are a very visible component of the fauna of corpses. Some adult Staphylinidae are early visitors to a corpse, feeding on larvae of all species of fly, including the later predatory fly larvae. They lay their eggs in the corpse, and the emerging larvae are also predators. Some species have a long development time in the egg, and are common only during the later stages of decomposition. Staphylinids can also tear open the pupal cases of flies, to sustain themselves at a corpse for long periods. Hyster beetles, family Hysteridae. Adult hysterids are usually shiny beetles, black or metallic green, which have an introverted head. The carrion-feeding species only become active at night when they enter the maggot-infested part of the corpse to capture and devour their maggot prey. During daylight they hide under the corpse unless it is sufficiently decayed to enable them to hide inside it. They have fast larval development with only 2 larval stages. Among the first beetles to arrive at a corpse are Hysteridae of the genus Cyprinus. Cyprinus adults feed on both the larvae and pupae of blowflies, although some have a preference for fresh pupae. The adults lay their eggs in the corpse, inhabiting it in the later stages of decay. Carrion beetles, family Sylphidae. Adult Sylphidae have an average size of about 12 millimeters. They are also referred to as burying beetles because they dig and bury small carcasses underground. 34. Both parents tend to their young and exhibit communal breeding. The male carrion beetle's job in care is to provide protection for the breed and carcass from competitors. Necrodes literalis is a type of carrion beetle in the Sylphidae family that prefers to inhabit bodies that are outdoors and in the later stages of decomposition. Modeling the activity and behaviors of carrion beetles like N. literalis and other carrion beetles that invade bodies earlier in death is a helpful tool to map time of death. 35-36. Ham beetles, family Claridae. Carcass beetles, family Troglodyte. Skin hide beetles, family Dermestidae. Hide beetles are important in the final stages of decomposition of a carcass. The adults and larvae feed on the dried skin, tendons and bone left by fly larvae. Hide beetles are the only beetle with the enzymes necessary for breaking down keratin, a protein component of hair. Scarab beetles, family Scarabaeidae. Scarab beetles may be any one of around 30,000 beetle species worldwide that are compact, heavy-bodied and oval in shape. The flattened plates, which each antenna terminates, are fitted together to form a club. The outer edges of the front legs may also be toothed or scalloped. Scarab beetles range from 0.2 to 4.8 in, 5.1 to 121.9 millimeters in length. These species are known for being one of the heaviest insect species. 37. Sap beetles, family Nitidulidae. Mites. Many mites, class Acari, not insects, feed on corpses with macrochels. Mites common in the early stages of decomposition, while Pteroglyphidae and Orobatidae mites such as Rostrosids feed on dry skin in the later stages of decomposition. Nicriforous beetles often carry on their bodies the mite Poecilocurus, which feed on fly eggs. 38. If they arrive at the corpse before any fly eggs hatch into maggots, the first eggs are eaten and maggot development is delayed. This may lead to incorrect PMI estimates. Nicriforous beetles find the ammonia excretions of blowfly maggots toxic, and the Poecilocurus mites, by keeping the maggot population low, allow Nicriforous to occupy the corpse. Moths. Moths, order Lepidoptera, specifically clothes moths, family Tinidae, are closely related to butterflies. Most species of moth are nocturnal, but there are crepuscular and diurnal species. During their larval stages, clothes moths tend to feed on mammalian hair. 39. They are amongst the final animals contributing to the decomposition of a corpse. This said, adult moths lay their legs on a carcass subsequently to fly larvae having had their presence on it. Wasps, ants, and bees. Wasps, ants, and bees, order Hymenoptera, are not necessarily necrophagous. While some feed on the body, some are also predatory, and eat the insects feeding on the body. Thus meaning they are parasitoids, parasitoid wasp. These Hymenoptera lay their eggs within the eggs or pupae of other insects, essentially causing the death of host insects. 21. Wasps can also be of association to the family Pteromelidae. The latter can lay single or multiple eggs. They oviposit in pupae of muscoid flies, blowflies. Subsequently, to the wasp eggs hatching, larvae will feed on the fly developing within the puparium, leading to its death. 21. Bees and wasps have been seen feeding on the body during the early stages. This may cause problems for murder cases in which larval flies are used to estimate the post-mortem interval, since eggs and larvae on the body may have been consumed prior to the arrival on scene of investigators. Insect succession. Life cycle of a fly. Insect succession, as utilized in forensic entomology, refers to the orderly progression of insect colonization and decomposition processes on a corpse over time. 40. Their life cycle typically consists of 4 stages: eggs, larval, pupal, and adult. Each stage has unique eating behavior that changes as the organism breaks down. In forensic investigations, the presence and developmental phases of insects can provide important data for understanding environmental circumstances, body movement, and postmortem interval PMI estimation. In forensic entomology, insects play a crucial role as indicators, helping to identify key components of a death investigation due to their distinct ecological responsibilities and varied geographic distribution. Given that different insect species inhabit a body in a certain order, understanding insect succession is essential for predicting the postmortem interval PMI. Typically, insect succession occurs in the following stages. A pig carcass in the fresh stage of decomposition. 1. Fresh stage, marked by the arrival of necrophagous insects attracted to the body by chemicals, such as flesh flies and blowflies. These insects deposit their eggs oviposit on or near to the body, and the decaying tissues are the maggot's food source. 41-23. 2. Bloated stage. The body starts to swell as a result of gas buildup during the breakdown process. During this phase, insects such as cheese skipper and coffin flies become more common. 41-23. 3. A pig carcass in the bloat stage of decomposition. Decay stage. In this stage, the insect community changes as the body goes through a more advanced state of decomposition. Predatory insects and mites increase in number, as do beetles, such as dermestid beetles and rove beetles. 41-23. 4. Dry stage. The body dries up and the skeletal remains become visible during the last phases of decomposition. Scavenger insects and animals may be present on the remains, along with insects like ham beetles and hide beetles. 41-23. Postmortem interval estimation. A pig carcass in the decay stage of decomposition. Insect succession, as utilized in forensic entomology, refers to the orderly progression of insect colonization and decomposition processes on a corpse over time. 40. Their life cycle typically consists of 4 stages: eggs, larval, pupal, and adult. Each stage has unique eating behavior that changes as the organism breaks down. In forensic investigations, the presence and developmental phases of insects can provide important data for understanding environmental circumstances, body movement, and postmortem interval (PMI) estimation. In forensic entomology, insects play a crucial role as indicators, helping to identify key components of a death investigation due to their distinct ecological responsibilities and varied geographic distribution. Given that different insect species inhabit a body in a certain order, understanding insect succession is essential for predicting the postmortem interval (PMI). Typically, insect succession occurs in the following stages. A pig carcass in the fresh stage of decomposition. 1. Fresh stage, marked by the arrival of necrophagous insects attracted to the body by chemicals, such as flesh flies and blowflies. These insects deposit their eggs oviposit on or near to the body, and the decaying tissues are the maggot's food source. 41-23. 2. Bloated stage. The body starts to swell as a result of gas buildup during the breakdown process. During this phase, insects such as cheese skipper and coffin flies become more common. 41-23. 3. A pig carcass in the bloat stage of decomposition. Decay stage. In this stage, the insect community changes as the body goes through a more advanced state of decomposition. Predatory insects and mites increase in number, as do beetles, such as dermestid beetles and rove beetles. 41-23. 4. Dry stage. The body dries up and the skeletal remains become visible during the last phases of decomposition. Scavenger insects and animals may be present on the remains, along with insects like ham beetles and hide beetles. 41-23. Postmortem interval estimation. A pig carcass in the decay stage of decomposition. A crucial component of forensic entomology is the calculation of the postmortem interval, PMI, which mostly depends on the observation of the insect activity on a corpse. 40. Using the life stages of insects discovered on or near a body, forensic entomologists can reasonably determine how long it has been since a person died. A pig carcass in the dry stage of decomposition. The pace of insect colonization and development is influenced by a number of variables, including its temperature, humidity, the presence of other living things and others. 40. The accumulated degree hour (ADH) approach, which determines the total amount of heat energy generated by a body since death, is one of the models and methodologies used by forensic entomologists to estimate the PMI. 40. Even through PMI estimation based on insect evidence is typically accurate, it is crucial to take into account additional elements including body position, ambient circumstances and insect behavior that may have an impact on insect activity. Factors. Moisture levels. Both the decomposition and insect activity on a corpse are significantly influenced by humidity. Elevated humidity can hasten the decomposition process by fostering microbial proliferation, which facilitates the breakdown of tissues. Additionally, the odors and gases produced by this microbial activity draw insects to the body. These smells are particularly attractive to insects like blowflies and flesh flies, which may quickly populate a body in humid conditions. 42. Low humidity, on the other hand, can impede the breakdown process. Mummification rather than breakdown may result from the body losing moisture more quickly in arid settings. Given that many insects need damp atmosphere to survive, this may discourage their activity. Nonetheless, some insects, like dermestid beetles, can survive in dry environments and can still be found on a body. 43-44. The existence of standing water next to a body can also affect the activity of insects. Aquatic insects, such as water beetles and some fly species, may be drawn to bodies near water sources. These insects can colonize the body and hasten its decomposition. Furthermore, because different insect species have distinct preference for habitats, the presence of water might influence the kinds of insects that inhabits the body. All things considered, the rate and pattern of a corpse's decomposition and insect colonization are greatly influenced by its humidity. To estimate the postmortem period and reconstruct the circumstances around a death, forensic entomologists can benefit from an understanding of how humidity levels affect decomposition. 45. Submerged corpses. Emily Goff, a noted and well-respected forensic entomologist, was assigned to a case involving the discovery of a decomposing body found on a boat half a mile from shore. Upon collection of the maggot mass, only one insect, Chrysomia megacephala, was discovered. He concluded that the water barrier accounted for the scarcity of other flies. He also noted that flies will not attempt to trek across large bodies of water unless there is a substantially influential attractant. In addition, the amount of time a maggot mass has been exposed to salt water can affect its development. From the cases Goff observed, he found that if subjected for more than 30 minutes, there was a 24-hour developmental delay. Not many more studies have been conducted, and thus a specific amount of delay time is difficult to estimate. 46. The main focus of a study accomplished by Payne and King, 47, using fetal pigs, was the insect